para que todos entendam. Depois a gente vai passar, então, como o idioma oficial da nossa apresentação de hoje será o inglês. As perguntas podem ser feitas em português, se alguém tiver dificuldade para fazer as perguntas em português, fiquem à vontade, que daí a gente faz a tradução. Vamos aproveitar esse momento da melhor maneira possível.
Aqui não está ainda.
Ficamos.
Agora sim.
É o famoso direito.
Não tem que repetir tudo. Quantos temos ao vivo?
Temos 4.
Bom, vamos começar no horário. Bom dia, pessoal. Vamos iniciar, então, agora a nossa transmissão do segundo dia de workshop. A nossa apresentação hoje será feita em inglês, com o professor Oliver, da Alemanha. As perguntas que vocês tiverem podem ser feitas em português, não tem problema. Se alguém tiver dificuldade, nós faremos toda a tradução. Eu vou, a partir de agora, começar. Vamos passar para o inglês a nossa apresentação, tanto a apresentação que farei do professor Oliver, quanto a apresentação que ele fará. E, por favor, fiquem à vontade para colocar as suas perguntas ali no nosso chat, como a gente fez ontem, que a gente, na medida do possível, fará aí tradução, respondendo as perguntas e tudo mais. Good morning, everybody. It's a pleasure to have today in our workshop Professor Oliver Gering from University of Göttingen, Germany. I met Professor Oliver during my PhD in Germany, and now he is a partner in our research group. Professor Oliver was from 2009 to 2017 in Michigan Tech University as professor, and since 2017 is chair of the Fourth Genetic Institute at the Georgia Goethe University, Göttingen. He has a very long experience with molecular markers, evolution, genetic of forest species, and for sure we'll have a great presentation that will help you or help us to have some more ideas for our research, to understand some new areas. And well, let's hear Professor Oliver. Oliver, thanks a lot for your disponibility to make this presentation. Please, we are hearing you, and people will make some questions, maybe in Portuguese, maybe in English, and I will try to help people here with translations and so on.
Yeah, thank you very much for your nice introduction. I hope you can hear and see me, especially hear me. I will give a presentation on microsatellites and want to give some little bit of history and background. And now I show you the slide. Hopefully that works out. Let's see. Okay, I hope you can see now my first slide. So I'm focusing on the application of specific genetic markers that are distributed across the whole genome, so-called microsatellites. So they are called microsatellites because they represent repetitive regions of the DNA, and they are actually present in different parts of the genome, not only in the nuclear genome, but also in the plastids. So in chloroplasts, for example, we also have like microsatellite markers that are used for specific applications. So why do we analyze these kind of markers? We would like to assess the level and distribution of genetic variation within populations. And since like the genetic variation is affected by neutral and adaptive processes, and actually the level of variation is also the prerequisite to adapt to new environmental conditions that we are facing in the future, it is of utmost important actually to understand and to characterize specifically populations with restricted geographic range and that might be isolated and face risks of extinction in the face of climate change. You are all aware of like biotic and abiotic stresses that tree species face. Here is a little bit of a perspective, European and North American perspective. On the right-hand side, you see the emerald ash borer, which actually killed a large portion of ash stands in North America. We also have in Germany now like ash dieback, but caused by a fungal species, and emerald ash borer is close. So in brief, like to adapt to these changes in environment and to adapt to stress factors, biotic and abiotic, like genetic variation is the basis. And you are aware that there are different ways to actually assess genetic variation. The costs for like whole genome sequencing or like genotyping by sequencing are decreasing, but still the cost per samples are comparatively high. If you are interested in specific genomic regions that show signatures of adaptation, then certainly you would have to characterize large parts of the genome and characterize these variants like either along environmental gradients or try to associate them with adaptive traits. But if you have specific questions related to like neutral processes like gene flow among population, the characterization of genetic variation, but also like in marker-assisted selection, if you have specific candidate genes that actually show also microsatellite variation. Microsatellite markers are very useful specific markers due to their high genetic variation and variability within populations. So there are a couple of different applications for which you can use genomic microsatellite markers. So they are not outdated, but still useful for many applications. And I hope I can present this to you in a little bit more detail. In the next slide, you see actually a sequence, and on the title, you see like the word simple sequence repeats, and actually that's what you see in the sequence. If you have a closer look, there is a short sequence motif that is repeated several times. And actually these repeat motifs, they occur across the genome, and they can be used to generate variable genetic markers because these microsatellite regions show high mutation rates. Maybe if you did like a polymerase chain reaction on microsatellite markers, you might have recognized like that the enzyme, so the Taq polymerase in vitro actually produces mistakes, so-called stutter bands, and also in vivo, like due to the replication process, like these mistakes occur more frequently than in other regions. So due to these high mutation rates, you can tag these microsatellite markers and amplify them via polymerase chain reaction. And for this, as you see, you need to know the flanking sequence of these microsatellite markers to design so-called primers or starter molecules that allow you to amplify this specific region and to visualize genetic variation in the number of repeats. And what you see is actually length variation, which corresponds to variation in the number of repeat motifs. So you can use these kind of sequence tag microsatellites for many applications, but as you see here, you need to know the sequence flanking these microsatellite repeat. And here is a real example. This is a study by a former PhD student in Göttingen, not advised by me, but by my colleague, Professor Finkeldey. The student is Madhav Pandey. I think, like Marcus, you will know him. And here's an example what we did. So we actually generated these sequences, and in the past, in quotation marks, in the early 2000s, so we couldn't really use like low coverage genome sequences, but we had to generate like libraries that were enriched for microsatellite motifs, and then sequence these clones and amplify them with flanking markers. So the program that is used, that's still available, Primer 3. So you put in your sequence, you highlight, you label the region of the microsatellite, and the program provides you with a primer pair that can amplify this locus, and the primer pairs are actually selected that they are specific enough that only one region of the genome is amplified, but also that these primers do not anneal with themselves or with the other primer, so that you really have specific amplifications of the gene locus that you targeted with the primer pairs. And another specific of microsatellite markers is that they are actually also, the flanking regions are variable, in a way variable that they are different among closely related species. So like microsatellite markers that are at random positions in the genome, they are quite variable in these flanking regions. So the transferability to related species is in some or in many cases relatively low. So they are very useful for many applications. However, you need like sequence information, and microsatellite markers, at least of the nuclear genome, have to be developed specifically for each species. And you see like a couple of examples. Traditionally, it was really time consuming to generate these enriched libraries for microsatellites. And then in later years, you can use like actually information from databases, genome sequences, sequences of cDNA, so-called expressed sequence tags. There you can use this information to use specific software packages to actually search these sequences for repetitive motifs, and then use again this program Primer 3 to design the specific primers. So why are microsatellites useful? This is briefly summarized again. They are hypervariable markers, so they are suitable to reconstruct descent. So you can use them, for example, for gene flow analysis in isolated populations. You see like gene flow within a stand, but you can also estimate how these populations are connected to each other by determining the percentage of genes that are introduced in the population. So another specifics of these nuclear microsatellites is obviously that they are codominant. So you can distinguish homozygous from heterozygous individuals. You see on the right-hand side an example from Norway spruce, and 6 selected trees were genotyped at one marker. And as you recognize, they show a high variation for different alleles. And if you count it, 1, 2, 3, 4 out of 6 individuals are heterozygous. So they also show a high observed heterozygosity, in this case of about 66%. Due to this high variation, they have a couple of applications, as I pointed out before, gene flow analysis, but also genetic variation, especially in species that are actually isolated and overall show low genetic variation. So the microsatellites show like length variation, as I pointed out before. This length variation is due to variation in the number of repeat motifs. And you see here in this example that the difference in allele sizes is always a multiple of 2. And if you look at this, you can already conclude that this marker is a so-called dinucleotide repeat. So a short sequence of 2 nucleotides are repeated xn times. So here a little bit of historic background. Obviously, you don't have to remember this in detail because now there are other options to use it. But if you outsource like kind of microsatellite development to companies, they still use like microsatellite enriched libraries. But now they are no longer, the PCR products are no longer cloned and then sequenced. They can actually sequence this using like high throughput sequencing method. But the general principle is the same. You isolate the total DNA of a species, and you cut this DNA in small pieces with restriction enzymes. And then you ligate adapter molecules, shown in yellow here, to these restriction fragments. And the next step is actually you fish out these microsatellite motifs using probes, hybridization probes that contain specific, frequently occurring motifs in plants. And actually these hybridization probes, anchor is actually ligated, so biotin molecules, and you can then fish these microsatellites out of the whole genome using like metal beads and magnetic separation. So that's quite of a nice technique. And you use like similar methods also in other genomic application, for example, in exome capture or similar ways to reduce the complexity of the genome. So that's the general principle. As I said, the next step is actually to clone these fragments in plasmids and then to sequence selected plasmids and develop these microsatellite markers. So you don't have to remember this in detail because the last 2 slides, there were more historical importance. But the first slide, this enrichment is still used, but you can now use novel sequencing methods to sequence these regions and then to develop the specific microsatellite markers. So here is then another example where we took advantage to develop microsatellite markers based on low coverage whole genome sequencing, and we had then 1 Illumina run and pooled a couple of different important North American hardwood species. So the coverage for each individual species, the average coverage is quite variable, but this coverage is still high enough to predict based on the sequences a relatively large number of genomic or nuclear microsatellite markers, GSSRs. But you see then also that then the usable markers are lower because you need also the microsatellite marker and then flanking sequences that you can use to develop the primers. And these numbers is then lower. This is the PAL or PAL, potentially amplifiable loci, are these loci that contain a microsatellite motif of defined lengths, but for which also flanking primers can be designed. So we were able to develop these microsatellite markers with relatively ease of experimental work, and there are ample microsatellites that you can use for gene flow analysis, and I will show you a few examples, and 2 publications are also listed below where we use these resources to develop and to test these microsatellite markers, whether they really amplify, whether they show null alleles, and whether they show high variation and easily interpretable patterns. So here is a little bit of a statistics for these different tree species pointed out earlier. you see the total number of usable microsatellite markers varies among species, but they are still like at least in the range of 1,000 potential marker, which is really largely enough for most population and gene flow analysis. You also recognize that these 2 base pair repeats, the dinucleotide repeats, are most frequent. Then there are like 3 nucleotide repeats and tetranucleotide repeats, which are less frequent. So you may recognize that these are randomly distributed. However, some of these microsatellites can also be located within expressed genes, even in coding sequences. And you will see in one later slide that this is actually the case. So in this study that I just briefly presented, we used like whole genome resequencing to develop these markers that are randomly distributed. And in this study here on European oaks, we actually used like sequence information of like cDNA. So this is actually transcribed messenger RNA. And these sequences contain like coding regions, but also untranslated regions, 3' and 5' untranslated regions. And you may recognize that here actually, as compared to a random set of microsatellite markers, the number of trinucleotide repeats is higher. And these markers actually are also quite frequently in the coding region of genes. However, there are not so many repeat differences. Do you imagine why actually trinucleotides are overrepresented in coding regions of the genome? I don't know whether you can answer it, but and whether I can hear you if you try to answer it. But actually, it's the mutations in trinucleotide repeats in coding regions. They result in one additional amino acid or the loss of one amino acid, but they do not disrupt the reading frame. So microsatellites also occur in coding regions, but they are actually generally trinucleotide repeats or potentially also hexanucleotide repeats. So these are other microsatellites. Since they are close to genes, they are generally less variable than like random microsatellites that are located in intergenic regions. So while like these microsatellites in intergenic regions are considered as selectively neutral, these microsatellites in or close to coding regions of genes, so-called EST SSRs, they could be under selection. The disadvantage is that these EST SSRs are less variable. Advantage is that actually the pattern is easier to interpret and that you can study specific potential signatures of selection at some of these markers. And another advantage of EST SSRs is since they are actually located either in coding regions or close to coding regions which are more conserved, which means these sequences are more similar or even identical among closely related species, the transferability of microsatellite markers that you developed, for example, in oaks, to other species is comparatively high. So we use a subset of these microsatellite markers, for example, also in European and Oriental beech, and I think it hasn't been tested for all species, but I think they also show a high transferability to other Fagaceae species. I forgot to say chestnut is also a species which actually markers are transferable to. So here on the right-hand side, I just have show you like so-called linkage groups. They show markers in a specific order located on the chromosomes of Quercus petrea and robur. And you see that you can also use these markers to align these specific chromosomal arrangement, the marker order, and to compare the marker order, for example, in oak or beech, on beech chromosomes to detect, for example, inversions or translocation. So they are also used for genetic mapping. So if you would like to design these kind of markers, there are a couple of programs that can be used actually to search these databases, EST databases, but also genomic sequence information to come up with a list of thousands or 10,000s of potential markers. And then you can use this information to develop specific markers that, for example, have an even distribution across the whole genome and are thus applicable to estimate, like in quotation marks, genome-wide genetic variation patterns. So here I briefly outline the fact that these EST SSRs can be used in different species. On the upper part, you see like the use of these EST SSRs that have been developed in European white oaks. They are applied to North American red oaks, to different species, and you can use them in that case to distinguish the species from each other, but also to identify putative hybrids. So individuals that show like this red bar, they are clearly assigned to Quercus rubra, and the ones that show like a blue bar are assigned to Quercus ellipsoidalis, and some of them, they have a percentage of ancestry, for example, of 40% in one species and 60% in the other species. They are putative hybrids. And some of the same markers were actually used and transferred to European beech, there to study actually also population structure within a species. And you see like that the structure changes along this graph from northeastern Germany to Switzerland. So again, like there are individual samples shown, we identified 2 genetic clusters and assigned each individual to these clusters. And you see some differentiation here between German and actually populations from Switzerland. So the point that I want to make here is, however, that you can use these resources as potential primers that can be transferred among related species, and that can be used for a couple of different applications. So, and finally, we talked about like randomly distributed genomic or nuclear SSRs, about EST SSRs, but microsatellite or simple sequence repeats are also located in the chloroplast genome. And actually for the chloroplast genome already a couple of years ago, and this is just one example, one paper, so-called universal PCR primers were developed for these microsatellite markers. So these microsatellite markers developed by Weising and Gardner, for example, they can be used virtually in all angiosperm species. So not all of the ones that are developed, but in the past, always a subset of these markers was usable to amplify in other tree species belonging to the group of angiosperm, so broadleaf species. So these markers can be, for example, be applied to study phylogeography, because like these chloroplast genomes in angiosperms, they are inherited via the seed, so maternally, so they reflect seed migration patterns, and there you can study, for example, in Europe, but also in North and South America, post-glacial remigration patterns of different species. The advantage of these markers is obviously that they are universal. These chloroplast genomes where the primer were designed, they are highly conserved. So the authors used like available whole genome whole chloroplast sequences to align them with each other to identify like the homologous regions, and they identified like regions that were identical across many angiosperm species, and in these regions these universal primers were designed. A similar set of universal chloroplast microsatellite markers is also available for conifers, but in conifers the chloroplast genome is paternally inherited, so via pollen. So in that case you can use chloroplast microsatellite markers to study gene flow by pollen or to assess like spatial genetic structure due to pollen dispersal. So here is just one example. As I pointed out, like oaks show a very strong phylogeographic structure. So like different geographic regions in Europe show different chloroplast types. And these chloroplast types on the left-hand side of the figure are just shown by different color. So each color is a different chloroplast sequence. And the pink color actually and the dark purple color, they originate like not from Germany exclusively, but they occur in the lowland of the Save River in today's Croatia. And the pink type exclusively occurs in Croatia, as you see in this left-hand side figure. So you can use these markers actually to identify like planted and introduced stands. And here we used actually these kind of chloroplast markers for these purposes to identify like introduced stands, but you can also use them for the certification of a reproductive material for their geographic origin. So this was just a brief, or maybe not so brief, but an outline about microsatellite markers and already some of the applications. And I would like to present like a couple of examples how you can use them to assess genetic variation and gene flow, differentiation between population, identification of varieties and species. Based on this, detect like putative hybridization and introgression events, and then briefly outline their potential use also in marker-assisted selection of specific superior phenotype or drought-resistant variants. So here just an example, the nuclear microsatellite that we developed based on low coverage whole genome sequencing. So this is actually is close to our garden in Michigan, so where we lived from 2009 to 2017. So we applied these, we developed specific microsatellite markers and used them for gene flow analysis. So like the whole stand close to our house was like genotyped at these microsatellite markers. And from a few selected trees, we collected also seeds. And adult trees and seeds were genotyped at the same set of markers, and using like parentage exclusion, we were able to identify like pollen parents within the stand. And you see like some of the neighboring trees contributed 8, 10 or 2% of the pollen, but the main pollen influx comes from outside. So even thought like this is a little bit of an isolated stand. Around the city there are many populations of sugar maple, and there is significant pollen influx, actually a typical pattern that you also observe in other wind pollinated tree species. So this is something for isolated forest fragments where you assume that there might be actually also effectively isolated because there is little potentially, there's a high distance to other conspecific stands. You could use this method to actually estimate like effective gene influx by pollen using these kind of paternity analysis. Within the plot, you recognize that there were like some neighboring trees also effective pollen donors. And that results actually in so-called family structures in natural tree populations. So family structures are due to restricted seed and restricted pollen dispersal in trees with heavy seed. Like this kind of spatial genetic structure is mainly due to gravity dispersed seeds. And here you see an example in European beech. Here the different genotypes at one specific microsatellites are shown in one stand of European beech. And you may recognize that these kind of gray shades are not randomly distributed, but there is some spatial clustering. And if you plot then the mean genetic distance in defined distance classes, you see that the distance between individuals in the first distance class is relatively low, and that the distance increases with geographic distance. And that's a reflection of like spatially restricted gene dispersal. So the dotted lines, they are actually the result of a permutation of individuals among distance classes. And you see here that you see like significant spatial genetic structure up to a distance of about 30 to 35 meters in this population, and similar results have been observed in other beech populations, but also in other actually wind-pollinated tree species. So this is not very extensive spatial genetic structure, but in isolated population that might look very different. For example, at the southern edge of the distribution range of European beech in Spain, spatial genetic structure extended up to 160 meter, meaning in this area or radius, like trees are closely related to each other, resulting then in the next generation also in like gene flow or effective gene exchange between related individuals, and in the long term, if these populations are isolated, also to inbreeding with negative effects on the survival of seedlings and adults, so-called inbreeding depression. We did some similar analysis actually in Afghanistan. So I haven't been there. The PhD student actually, he is from Afghanistan. Before moving to Germany with a DAAD grant, he collected individuals from adult trees of Pinus gerardiana, so an important species not only in Afghanistan but also Pakistan. But the natural regeneration is largely missing because of overutilization. And in that case, we also assessed like the spatial genetic structure, and you see a similar pattern. Actually, it also extends to about like 20 meters. So there is no significant spatial genetic structure, which is good news in our case. Also, the genetic variation was comparatively high, but the problem is really the near absence of very young natural regeneration, the population is okay right now, but future generations, that's problematic because natural regeneration is lacking. This is also an example from Professor Stevenon during his PhD students in Araucaria angustifolia, where he used like microsatellite markers and genome-wide AFLP markers, and a very similar pattern is observed. But also an important observation was made that we also made now when we use like genome-wide sequence information to assess spatial genetic structure, that the more marker you use, like you have a higher power to detect actually this fine-scale genetic structure. So now I'm coming to some examples how we use these markers to analyze genetic differentiation between populations, varieties, and species. This example you have already seen. And I will just briefly outline this. So you can use this to study genetic variation within and among populations. at microsatellite markers since they are highly variable, but also because, like most tree species, have very effective means of seed and pollen dispersal. Most of the variation is distributed within populations, but using a larger set of markers, you can still detect spatial genetic structure. You see, like again, like the German populations are dominated by the blue color, and the populations from Switzerland are dominated by the orange color. So we have like large scale geographic structure, and you see on the upper part of the figure where we analyzed like the data, again assuming that there are 4 genetic clusters, that one of the German populations, AEW8, is also differentiated from the other ones. So apparently it has some other geographic origin. We also looked at a large number of other German population, and this population AEW8 somehow sticks out, is different. And it's also different when we look at the growth performance in the Common Garden experiment. It shows a higher growth performance at 3 different sites in Germany. We still don't know where this population actually originates from, because it doesn't fit with the other reference populations clearly. So another example I also outlined before, like identification of like provenances from other geographic regions. And this is the Slavonian oak. Actually, it has been intentionally introduced into Germany because it grows really fast. It has a nice trunk shape, not many branches, and is really late flowering and kind of seems to be well adapted to some conditions in the lowlands of Western Germany. And some of the stands are actually known, their origin is known and others not, and we used like genetic markers to confirm or to validate their origin. And on the left-hand side, you have already seen this. Actually, all the circles represent like populations or stands, and the ones that are in pink, they are for sure from today's Croatia, so they are Slovenian oak, because this specific chloroplast variant or haplotype does not occur naturally in Germany. The other like dark purple one actually is predominant in Croatia, but also occurs in Germany. There we do not know, so we used then actually again like nuclear microsatellite markers. On the left-hand side, these were the microsatellite markers. On the right-hand side, nuclear microsatellite markers. And then you see that actually the ones that show the chloroplast variant that occurs in Germany and in Croatia, based on chloroplast microsatellite, it groups with the Slavonian oak. So with this combination of set of markers, we can identify like the relative proportion of like Slavonian oaks that are supposedly later flushing and faster growing than autochthonous indigenous varieties, using these kind of set of genetic markers. And we did this not only for adult trees. You see like here these Slavonian oak stands, the adult trees labeled here, and on the other side, NR is natural regeneration. Natural regeneration is actually has been collected from the ground underneath these adult stands. And if you compare these patterns, then actually it looks quite similar for adult trees and neighboring Slavonian stands, which actually means there is little gene flow, even though like indigenous oak stands are close by and in majority, but as I pointed out earlier, Slavonian oaks are late flushing, so apparently due to these differences in flowering time, apparently which is little overlapping, there is little gene flow between like these introduced and indigenous varieties. Okay, another example here, I make it a little bit shorter. Here we used microsatellite markers, again chloroplast microsatellite markers, to analyze introduced northern red oak stands in Germany. So they have been introduced already in the 17th century to Germany, also because they first in parks, but now also because they show a high drought tolerance. And here we use these chloroplast markers to assign a putative origin of these German provenances. And then we recognize the chloroplast haplotype in green, which actually only occurs in the northeasternmost distribution range of the species in North America. And this haplotype, this chloroplast variant, is not frequent, but it occurs in many of the stands and populations in Germany, indicating actually origin in the northeast of North America. Now we have additional chloroplast markers identified that actually confirm this result. So this is another example where we can use like specific, as I said, universal markers, but here we also developed based on chloroplast genome sequencing. We also developed like oak specific markers to actually have a higher resolution of chloroplast haplotypes that allow to assign the geographic origin of selected samples, in this case of introduced German stands from their geographic origin in North America. Because like these microsatellite markers can be used actually also to assign individuals to species, if we identify like these markers that show like sufficient differentiation between these taxa, we can also identify like genetically intermediate individuals. And that's also useful to assess the level of the level of introgression between closely related species, either naturally co-occurring, but also of like introduced populations of like different subspecies, because that it might be beneficial. I mean, the introgression of new genetic variation into the species for like rapid adaptations, but it might also result in outbreeding, depression, and lower survival of the outbred offspring. From a nature conservation point of view, it's also of importance whether there is actually gene exchange between introduced and natural species. And here we looked at Fagus sylvatica, European beech, and Fagus orientalis, which is actually planted in Germany and which is considered as an alternative to Fagus sylvatica in the future if like drought events are more frequent, but little was known actually about like gene flow between these two species. So there we first looked in the natural range and actually again used microsatellite markers to characterize like Fagus sylvatica populations and morphologically identified Fagus orientalis populations. And as you see here, using these genetic assignment analysis, again, FS stands for Fagus sylvatica. So apparently this is assigned mostly to this blue cluster, but there are also potentially, especially in the contact zone between the two species in Greece, there are intermediate forms. So there is a hybrid zone between the two species, geographically restricted because the ranges of the two species or varieties are only little overlapping, but that also indicates that there is also potentially, or it's quite likely that there is also hybridization between introduced Fagus orientalis stands in Germany and native Fagus sylvatica stands. And here you see on the lower hand, we were able to quite efficiently separate these two varieties from each other because like there was one microsatellite markers, and this is actually a marker located in a gene. So an EST SSR marker, a trinucleotide repeat, which actually quite efficiently differentiates between Fagus sylvatica and Fagus orientalis. You see the allele 189 is predominant in Fagus sylvatica, and the allele 192 is predominant in Fagus orientalis. So using these kind of markers actually in one pilot study, in one population that at least among German foresters is quite famous, in this so-called Erdmann forest in northern Germany, we genotyped actually Fagus sylvatica and Fagus orientalis, adult trees, and they were quite well separated from each other. Sylvatica, you see all individuals grouped with this in this blue cluster, and orientalis in the orange cluster, but there were also hybrids, and these were mainly younger plants, so either seedlings or young adults, so the second generation. So there is apparent hybridization actually in these in the natural regeneration of this Fagus orientalis stand. Looking at some more detail, and here we used a combination actually of chloroplast microsatellite markers. You remember this marker CCMP. These are the universal chloroplast markers described by Weising and Gardner that work for many angiosperm species. With this marker, actually, we can distinguish Fagus sylvatica from Fagus orientalis. It's uniparentally inherited, so you can identify like the seed parent. And the individuals that have the marker allele 152 originate actually from Fagus orientalis as a seed parent. So the natural regeneration has Fagus orientalis as the mother. And the individuals that are characterized by the allele 145, they have actually Fagus sylvatica as the mother. And by looking at this pattern, there are interspecific hybrids for like Fagus orientalis mothers and for Fagus sylvatica mothers, meaning that the gene flow between the two species is bidirectional. Since like Fagus sylvatica is more frequent in the region, due to this abundance, also like gene exchange from sylvatica to orientalis occurs more frequently. And here is just the same from a published paper that again like specifies that in these very high DBH classes, so trees with high diameter at breast height, these adult trees are clearly assigned to species orientalis and sylvatica, and in the lower diameter classes there is a larger abundance of actually genetically intermediate individuals, so these vertical bars that show both green and yellow color. Okay, now I hurry up maybe a little bit. Another example from North America where we lived before, like the two closely related species, Quercus ellipsoidalis and Quercus rubra, that co-occur in contact zones on very dry sandy soil. And we used like, again, microsatellite markers, a randomly selected set to identify the species and to identify putative hybrids, so that these individuals that show like this, whatever it is, pink and blue combination of colors. And then we also recognized, even thought with reference sample, they can be identified as genetic distinct entities. So their status as different species was confirmed. The interspecific differentiation was quite low. It was like on the range of 4 to 5%. So most of the variation is actually within species, and the interspecific differentiation amounts to just 5%. But still they remain morphologically distinct, and also distinct in terms of their ecological preferences, since Quercus ellipsoidalis shows a higher drought tolerance than Quercus rubra. And when we looked again at each individual markers, and that's actually related to marker-assisted selection or markers that are potentially under divergent selection, we genotyped like about 50 EST SSRs, which are located in the coding region of genes or close to the coding region of genes. And we assess the interspecific differentiation for each of the markers. On the y-axis, you see this interspecific differentiation, so-called FST. So that's just the percentage, the relative differentiation between species relative to the total variation. So for most of the markers, the differentiation is relatively low. So on average, as I said, 5% of the variation is distributed between the species. And then you see one marker. First I thought it's a mistake, and that's a nuclear microsatellite marker that shows a differentiation more than 80%. So it's not fixed, but it's nearly fixed on alternative alleles in the two different species. And that's a gene with a putative function in Arabidopsis in the control of flower initiation, flowering time, and growth cessation in late summer or autumn. So that might actually contribute to the differentiation between the species. Flowering times are not overlapping. So while most of the genome is homogenized by interspecific gene flow, some of the genes resist this gene exchange as a result of natural selection. And these kind of markers are not neutral, but they have a putative role actually in adaptation of the species to their environment. And this kind of selection seemed to be different across the range. So we just analyzed like 3 different populations close to Lake Michigan and Lake Superior, and assessed like interspecific differentiation between these populations. The first example, the differentiation is higher than 80%. However, in the other population pairs, the differentiation is only 64 or 38%. And you see also on the right-hand side that the soil conditions are different. So the soil contrast is less pronounced actually in the two populations on the bottom of the figure. So indicating that the environmental conditions also have an effect on the introgression of these specific genes, so-called outlier genes between the related species. And when we looked at this in more detail in the so-called sympatric stands, where actually both species co-occur next to each other on very dry sandy soil, which is not characteristic otherwise for Quercus rubra, we see actually based on the frequency of the species diagnostic allele that this introgression is not symmetrical, but that more of these genomic regions involved in drought tolerance are transferred from the drought tolerant species Quercus ellipsoidalis to the more drought adverse species Quercus rubra that has been found in 2 of these examples of sympatric stand, but that's not the case actually where they do not occur in sympatry. So now I'm really coming close to the end, talking about these marker-assisted selection, but not so much the use actually of microsatellite, but the use of microsatellite as a neutral reference. Since like if we want to find like markers that are associated with environmental differences or with different morphotypes, as here shown for high and low elevation spruce trees, so we need to account also for the neutral population structure. And we did this in these spruce varieties. On the right-hand side, this is close to Göttingen, about 2 hours in the mountains, Thuringia. And there are these high elevation spruce trees that have like pending low-hanging branches as an adaptation to heavy snow load. We wondered whether this is a different variety and used like microsatellite marker assess a neutral genetic variation, but I just summarized the result. There is no differentiation between high and neighboring low elevation stand, However, there is some geographic structure, so the populations from one mountainous regions, the Harz Mountains, are differentiated actually from the other stands, and otherwise there is not a clear, consistent pattern. If we use like this neutral genetic variation as a background, we can try, or we associated candidate genes for adaptation to latitude, altitude, and freezing temperatures actually. So genes that were actually associated with latitudinal gradients to compare their pattern in the low elevation stands that potentially are not adapted and to the high elevation stands. And we identified a couple of these candidate gene markers, and actually candidate gene markers potentially playing a role in the local adaptation of the high elevation stands to specific conditions. And these are genes also involved in phenology, so the timing of bud burst, but also like bud initiation in late summer and autumn, so growth cessation. So these markers, as I pointed out earlier, can then also be used to calculate genetic linkage maps to associate then really traits of interest like growth performance, drought tolerance, stomata density, a couple of different traits with like genome-wide genetic variation. And for this purpose, we perform controlled crosses to generate full subfamilies that segregate for genetic markers, but also for traits of interest, mainly traits of phenology. And we use these genetic markers to align these kind of linkage groups or chromosomes with each other. And here you see again that the microsatellite markers that were used, for example, GOT stands for Göttingen because we developed this here. So this marker actually amplifies in Robo and Petrea and can be used to align these chromosomes with each other. But also you see that the same markers amplify in chestnut and in oak, and these markers also can be used to align the chromosomes to each other. So this information can then also be used because we have like compare homologous genomic regions to identify genomic regions that are associated with traits of interest. And really interestingly, phenology, the timing of vegetative bud burst actually is controlled by a similar genomic regions in oaks and in chestnut. So these kind of comparative mapping approaches can also be used to identify candidate genes, and now with actually availability of genome sequences, also the candidate genes can be identified in these genomic segments. So here I thought that I'm a little bit faster, but here I'm actually at the end. So this is our group working on different aspects of population genetics in Göttingen, close to a little castle, Burg Plesse. Professor Stevenen will remember. So these students, postdoc, and people did the work, and I just presented a little bit. So I'm then here at the end. This is just a view in Michigan, just where the red oaks have been sampled. So thanks a lot for your attention, and yeah, I'm open for any questions.
Oh, thank you for your presentation. It was very great and interesting. That some things that I told people yesterday about the characterization of markers and some aspects related to the development. Now you brought us examples of using this, for instance, for these EST markers, and it was very nice because it's complemented all the presentation.
Oh, thank you. Oh, that's good.
Pessoal, alguma pergunta?
Microsatellites are still quite useful. I mean, there are some people saying you should do like whole genome sequencing, but that's not affordable. And for like targeted analysis and also for neutral variation patterns, it's very useful.
And I think that maybe for the so-called major crops like maize, beans, and so on, it maybe is more useful or easier to use the whole genome sequence. But when you think about some species like endangered ones or such economically less exploited species, it's still too expensive. Although the next generation sequencing is cheaper than it was some years ago, now it is expensive for many labs. So microsatellite is still very useful.
Yes, yeah, I agree. Yeah.
And so while people is thinking about the question, I will make one question. You showed us here this use of cross amplified markers in Castanea and Quercus for QTR identification and so on. Consider that today is relatively easy to sequence a genome, relatively cheaper. What do you think about the test of cross amplification of markers for genetic studies, genetic diversity? Do you think it is still useful, or no, we will not more do this kind of tests and so?
Yeah, I mean, for us, it's useful because also we work with a number of species where there is little or no information. So we usually also test like markers and their transferability to closely related species and apply this then for population genetic studies. So it's, I mean, maybe if you have the funds, it would be, if you have a number of target species, I think it would be useful maybe to use something like low coverage whole genome sequencing to develop this set of markers for future studies. But we often also cooperate like with a wide variety of countries, a wide variety of species. It's more like driven by the project, by the interest in the project. So, but I would say if you have a number of target species that you would work on, I think it might make sense to use this kind of sequencing and to develop a new set of markers. So we're quite happy with chloroplast microsatellite markers because there you also with species with no genomic information, you can do something. That's very, very useful. And EST SSRs, Fugazi is something specific. It can be transferred across different genera. In most other cases, it's maybe only among species within genera. It's quite different. Yeah, I would consider it as useful. I don't know whether it's to use transferability tests.
Ele consegue ver a pergunta lá?
Sim.
So we have a question from Juliana. Juliana is a student for our university here. I think you can see the question.
Yes. Thanks a lot for the question. I think everybody can see it. Yeah, related whether hybridization and introgression could result in positive effects related to climate change. I was speculating that might be the case because there is like asymmetric introgression of drought-related genes in very dry environments. So it's a potential mechanism, at least in oaks, to rapidly increase genetic variation at adaptive genes where they co-occur. So potentially that's a mechanism. To test this, and nobody has done it, but I think it would be nice to test it. It would be good to have like seed material from putative F1 hybrids, to plant them in the greenhouse and then to actually assess the drought tolerance of these seedlings with different levels of introgression from the other species. So, but it could be a way to rapidly increase the level of genetic variation. There is no clear evidence in oaks that there is very strong outbreeding depression, but it hasn't been clearly experimentally shown. No kind of intrinsic crossing, or no clear evidence for intrinsic crossing barriers. And for the Fagus orientalis, also Fagus orientalis is supposed to be more drought tolerant than Fagus sylvatica, but it's also a problem. I mean, it might be that like hybridization with Fagus orientalis increases drought tolerance of the seedlings, but it also kind of like generates kind of a new hybrid swarm. And yeah, I think it should be handled maybe with caution, but it could have positive effect, especially if we consider these scenarios like of very significant increase of temperatures. But if that really comes true, maybe we need other species, I'm not sure. But it's hard to say because these models, I mean, I think they do not necessarily consider topography and things like that. But yeah, briefly, yes, that could result in an increase of adaptive genetic variation. But it needs to be experimentally tested how valuable this approach would be. But I would see that more like in oaks that naturally co-occur, actually to also promote the hybrids, because from a forestry point of view, hybrids are not good. They have to be the one or the other species, and hybrids are not very highly valued. But doing this kind of research might, and considering climate change, might change this point of view a little bit, hopefully. Yeah, I don't know. I talked a lot. Hopefully it partly answered your question.
O pessoal está agradecendo ou não está fazendo
Só agradecendo.
Well, while people are thinking about the question, I will point another aspect about the usefulness of the microsatellite markers. We tried, I think, 1 or 2 months ago. We are talking with people from France, and they proposed us to work with SRSEC. That it means that we need to have the much more information about sequencing, or at least much more SS markers.
Yeah,
that are in Europe. How you see the movement? People are, I'd say, moving from microsatellites to genomics, to whole genome sequencing, local variation, or the movement is, okay, let's work with both, with traditional marker, PCR, and electrophoresis, and so on, and also with whole genome sequencing. How is the movement of the researchers, of the institutes, and so on?
Yeah, I would say both. For like genome-wide variation pattern needs to be analyzed if you want to do like environmental association analysis, or in provenance trial, genome-wide association analysis to identify like markers or gene candidate genes associated with the traits of interest. So I don't know whether it's really the future, but right now for us it's still efficient to do these kind of genome-wide association analysis to identify like interesting candidate genes for one specific target marker. And then we also use like targeted SNP genotyping using our mass array genotyping system for these. For these kind of environmental association analysis or association analysis, microsatellites I wouldn't necessarily use. but to assess like population structure and genetic variation, I think they are still like in terms of cost, the marker of choice and the applicability. For gene flow analysis, I think they are very useful and also still like, yeah, for genome sequencing, I don't know, still they have been useful to generate these linkage maps. And I mean, now with long read sequencing, maybe that's less of an importance, but still they are used to assemble the genomes. Yeah, I think both is useful. We still also, when I started, I applied for a new capillary sequencer since I think it's useful to use these markers. Because you are more flexible also. I mean, if you have to pay 60 to 100 euro per sample, you need a large grant and we need to outsource it. And the microsatellite markers, you really are a little bit flexible to do like some pilot project and you can decrease the cost when you work more efficiently in the lab. But if, yeah, you have more under your own control, I would say, if you have this genotyping still in-house and use this kind of marker.
I agree with you. Really. Really, budget is still the problem for all the people. So we have to plan all the steps, because maybe we will need a lot of money, but even though we cannot answer our questions, so it's necessary to plan exactly. And not all the really new techniques will be so good as the old ones, let's say. We are also using here, for instance, the plastic, the CP-SSRs for, we are creating a database for pecan nuts, because it's a North American species, but here in South Brazil, there are many people planting, and we are actually the fourth producer of pecan in Brazil.
Okay.
So we have
very
a
tasty.
large producer. The problem is that people lose the country, so they sell the nuts for one cultivar, but they are not sure if it really is this cultivar. And for instance, people from China intended to buy the nuts from Brazil, but they want to know, okay, which cultivar we are buying. They cannot be sure about that. Sometimes the crossing occurred, and people saw that this plant is better for against some disease or more productive, and selected these three. But now we don't know which one is this. So we're using the CP-SSRs to try to identify the crossing, or so, which are the parents of these species, and to make a kind of database. Actually, we are using CP and nuclear markers to create this database to certify. So this is cultivar X, Y, or so on, Barton, Success, and so on. And really, these markers are quite useful for such certification. We can also use for breeding, of course, to select these plants as marker seed selection.
Yeah, I think that's very useful, but that's not necessary to generate whole genome-wide data. I think it's, they have gene banks or cultivar identification, and I think also clonal identification, microsatellites are excellent, spatial genetic structure, also gene flow. Yeah, I see also, I do not agree with some of my colleagues that should be outdated.
Me too. So people, there are more questions, please. This year in June, my student here, Johan, will stay a short time in Germany. Dr.
Good.
Oliver will receive Johan, and now it's all right to Oliver, but we will talk about the end of this year, next year, to bring Dr. Oliver to stay for a short scientific period here with us. We just have to talk to see the better moment. I think that you should come here in our summer period to
That's
see
a good idea.
our sun, because I lived in Germany and I know that the sun is not so frequent in Germany as it here.
That's right. Yeah. In the winter, it's really a bit depressing. The summers are getting better, but too dry. That's strange. But yeah, would be a good time in the winter. Yeah, there we can. I would love to come, and maybe we can. Yeah, I don't know. There will be some maybe DAAD or Erasmus. I'm not sure. I have to look into this maybe. Or I can also finance from our household money.
I have some budget from my project, so for a short period, I can bring you without problem. But if you get some more possibilities, it would be nice.
Yeah, I
But we'll
think
talk
about it.
about this and make all the bureaucratic steps
Yeah.
to bring you for sure.
No, I would love to come. Yeah, I've never been to Brazil and also not to South America, but I would love to visit,
So
especially you. But it would be also new for me as a
yeah. No, it will happen.
destination. Oh no, this is excellent, and yeah, and obviously you are also invited to Göttingen if you maybe we
Oh yeah,
find some option also.
I tend to go back to Göttingen. I miss Göttingen, really.
No, it's actually quite nice, but yeah, in the summer also you should come in the summer or spring or Christmas market, but
Oh, Ingrid is my student.
thanks a lot. That's very kind. Yeah, it would be also a pleasure for me to come.
So, people, if you don't have more questions, I think we can finish here. Of course, Professor Oliver has much more things to do, and we will continue here. If there's no questions more, so Oliver, thanks a lot. It was grateful. Uh, I hope that we have more such moments for this knowledge interchange. If you think that we can help for some event here also in Germany, please contact us. And thanks a lot. I hope that we have much more moments like this in the next month through this project that we are working together and through other projects.
Yeah, I'm looking forward to this. Thanks a lot. And if there are any other questions, yeah, just drop me an email or we can. Yeah, we stay in contact anyway. But if your students have questions, they are welcome to contact me by email. No problem.
Right. Ok, pessoal, então nós vamos finalizando, certo? À tarde nós retornamos a partir das 14 horas com a palestra da professora Peggy Almair, da Universidade de Misiones, na Argentina. Tá? Se vocês tiverem aí alguma pergunta, quem for com vergonha de perguntar alguma coisa para o professor Oliver, pode nos fazer aí a pergunta, nos enviar, que a gente faz o contato. Temos o contato direto com o Oliver, então, eventualmente, se alguém ficou com alguma dúvida ainda, ou está formulando as suas perguntas, fiquem à vontade para nos contactar e, posteriormente, a gente entra em contato com o Oliver e responde as perguntas que possam vir, certo? Nos vemos à tarde, então. Ok.
Bye.
See you. Bye-bye.
Bye.
Ele ficou bonito.